Hello everyone and welcome to tutorial 1 of BChem 443. I'm Colin and today I'll be taking you through a virtual experiment for detecting glucose consumption in red blood cells when infected with the malaria parasite. Please feel free to pause this video along the way to see the slides that I talked to too quickly. It's important that I mention that all numbers and data in this video are for example, and the data to be used for the assignment can be found on D2L as an Excel document. So we have some learning objectives for our first tutorial here. We have 3 of them. We'll be learning how to solve problems related to human health. We'll be troubleshooting issues in experiments, and we'll be relating physiological changes to experimental data. So a bit of background on malaria. There'll also be a video lecture uploaded this week on malaria, so if you want more information, you can watch those as well. So it's an intracellular parasite called Plasmodium falciparum. It's transferred by mosquitoes and lives within human liver and red blood cells. And the red blood cell stage is often the target of research studies. Resistance is also common in the malaria parasite to common therapeutics. So what we're looking at today is the glucose consumption as a source of energy of Plasmodium within the red blood cells. Glucose enters the red blood cell, is transported into the parasite cytoplasm, and utilized by glycolysis and the pentose phosphate pathway in order to replicate rapidly within human cells. So we have a hypothesis that's twofold for this experiment because we're going to be looking at it in 2 different ways. We'll be looking at the glucose consumption by the parasite, and we'll also be looking at the possibility of new treatments. Our hypothesis, we will be first looking at Plasmodium infection and causing a deviation from normal glucose consumption in red blood cells. Secondly, we will be using this consumption change as an indicator for testing effectiveness of new antimalarial treatments. So to do that, today we will be using enzyme assays. So we'll be using them in 2 forms, a glucometer and a 96-well plate absorbance assay. The enzyme assays measure enzyme activity, and in this case, our enzyme activity will be for glucose oxidase. So to track the consumption of glucose, we will be using the glucose oxidase enzyme assay. It oxidizes glucose into hydrogen peroxide and gluconolactone with an FAD cofactor for the enzyme. The mechanism for the enzymatic reaction is pictured on the slide above. The FAD cofactor is very important. It's reduced by the glucose molecule in the step creating gluconolactone. It then generates hydrogen peroxide from oxygen in the next step, and then it regenerates FAD to be used as the cofactor throughout the rest of the reaction. So this reaction by glucometers uses the peroxidase enzyme for detecting how much hydrogen peroxide is being produced. It allows for a colorimetric change based on this production, and the use of glucometers have different applications in human health. So now we'll have an interview with an expert, Dr. Mohammadi. He's the lead engineer in the Lewis Research Group studying the use of biosensors in human health. He will be giving a background on how glucometers work, and as well as other biosensors, and this video is just for your guys' interest.
My name is Mehdi Mohammadi, a postdoctoral researcher and lead engineer at Lewis Research Group, University of Calgary. Different type of biosensor have been developed over last 20 years, from optical sensor, physical sensor, and electrochemical sensor. And today I want to briefly talk about electrochemical sensor, which divided in 4 main groups for detection: amperiometric, voltammetric, potentiometric, and impedometric measurement. And so the current glucometer in the market is based on the enzyme-based glucometer that's basically categorized in amperiometric, and divided in 3 main groups. The first group that has a glucose oxide on the strip, and the glucose oxide oxidizes the glucose and yield to gluconic acid and hydrogen peroxide. So measurement of the hydrogen peroxide indicate the level of glucose in blood or urine. And the second group use a redox mediator like ferricin, which interact to glucose oxide and increase the ion current and the sensitivity of the sensor. The third group actually has engineered enzyme and nanostructure electrode coated with a nanocarbon tube combined with the glucose oxide to increase the level of the sensitivity and the specificity of the sensor. But mainly the commercial glucometer in the market is categorized in group 1 and group 2. Also, there are some non-invasive electrodes that working with sweat, tears, and saliva in the market. And 4 or 5 main components of the electrochemical sensing is analyte, biorecognition section, and the transducer electrode and display. And the electrode itself divided to 3 components, counter electrode, reference electrode, and working electrode. So here in the Lewis Research Group, we have great potential to develop these electrochemical sensor to target any targeted biomarker in blood or urine or any biofluid. And also we could integrate to the tiny microfluidic channel for point of care application. And yeah, that's it.
For the first part of the experiment, we will be monitoring this glucose consumption over time with infected and uninfected red blood cells. We will be monitoring the change in consumption using the glucometer. So first, we need to generate the calibration curve for the glucometer, and these videos will take you through the steps.
We're going to go over our glucometer calibration curve. Here we're making the standards for the calibration curve. Due to the low mass of the product that we need in our calibration, we're going to be using this small Eppendorf tube directly in our weighing scale. We're going to put it on, we're going to zero it, and then we're going to take our product, weigh it out as close as we can to our calculated masses, and then we're going to go back and adjust our dilution to make the proper volume and concentration that we need for our standard curve.
So here we are weighing out the glucose standard for our calibration curve. We have a microcentrifuge tube resting on some tape within the scale. That way the glucose is measured directly into the tube for the measurement. We never take any powder back out of the tube, and we try to get as close to the mass that we've calculated, and instead of adding more or taking out more, we adjust our dilution volume to get the proper concentration for our standards. So you want to read your mass and then change your dilution after to get the proper concentration.
So at this point, we're going to read our glucose concentrations with the glucometer. Once again, we need to go through our standard curve with the glucometer first before we get into our infected and uninfected cells. So what we're going to do is take our P20 pipette, get a small amount of our dilution, then we're going to place a dot on a sheet of parafilm so it forms a nice round ball for us to actually use. Here's our glucometer. What we're going to do is place the test strip onto the ball, and then the glucometer will give us our reading, and then from here we're going to make our standard curve and compare it to our uninfected and infected red blood cells.
So first, this data here is given to you in the Excel sheet on T2L, so you don't have to copy it off this slide, just work directly off the Excel sheet. So why do we make a calibration curve? Our glucometers are meant to read glucose in human blood, not in media. So we need a calibration curve to get an accurate measurement of glucose in media. We have 4 replicates of our curve measurements, and you will need to take the average of these and compare it to the known standard concentrations. This data will be used to make figure 1 of your report, and make sure you include the equation in the figure caption of your report. So again, this data is also in the Excel sheet, so just work off of there. Here's the glucose consumption data for uninfected and infected cells over time. These will be used to generate figure 2 and figure 3 for your report, 1 for each of the treatments, and include the equation in the caption. From these figures, you can find the rate of glucose consumption in millimolar per hour. So now we are going to use a 96-well plate layout and the glucose oxidase assay colorimetrically, rather than measuring each individual well with a glucometer, which would waste materials and time. So we want a high throughput method this time. We use the glucose oxidase assay as an indicator for the effectiveness of a possible antimalarial drug. The red blood cells were incubated for 24 hours with plasmodium and a 5 millimolar starting concentration in each of the wells. And they also had 1 of 16 possible drug candidates. This assay relates the absorbance to concentration using a standard curve. The videos walk you through the steps. On the next slide here, I will just have the plate layout, which is very important. The plate layout will help you work through the data. You need it for generating your standard curve as well as the drug target results. Each therapeutic agent is replicated 3 times, and the assay measures absorbance based on glucose oxidase activity. And remember, it's very important in the starting glucose concentration in the wells is 5 millimolar. Our positive control has infected red blood cells. Our negative control has uninfected red blood cells. And our standard curve is replicated 3 times, and the concentrations are shown on the screen. So these videos will just walk you through the steps.
So now we're going to do a high throughput method to test for our therapeutic agents and whether we get the results that we're expecting for infected red blood cells. So now we would take our samples of interest, load them into our wells. At this point, they have a standard glucose concentration that we know in the media. What we're looking for is a change in this glucose concentration. In our plate here, to be read, we have 3 replicates of our positive infection control, 3 replicates of our negative infection control, and the standard curve using the same standards that we made up for our glucose measurements using the glucometer. All the other wells are filled with infected red blood cells with a known glucose concentration that we will be adding a new therapeutic agent to and testing for the new glucose concentration that we see in the wells.
So here we are adding our 3 replicates of each of our positive control, our negative control, as well as 3 replicates of the complete standard curve, and 3 replicates of each of our different therapeutics which we're testing for. It's important at this step that you don't introduce bubbles which would interfere with the absorbance readings. You will only want to go to the first stop on your pipette. That way you're not going to be releasing air into the wells which could introduce bubbles and interfere with your absorbance reading. Here, after you've done all your hard work, you want to remember to label your plate. Make sure you do this so that nobody throws it away when you're in the lab, losing all your hard work. Then we'll be measuring the absorbance of our 96-well plate in our absorbance reader. It's computer controlled, so you want to open the reader and then you'll insert your plate. When you insert your plate, make sure you remove the lid because that could also interfere with the absorbance readings. And then close it, get your absorbance readings, and then you have your data which you can make your conclusions based off of.
Use the data provided on the D2L Excel file to make your final figure, which is a standard curve. Remember to include the equation of the figure in the caption. Use the data to make a conclusion as to which therapeutic is most effective. And you can use the standard curve to find the final glucose concentration of the most effective therapeutic.